We are recording. We still have about 5 minutes to go. However, you are currently watching, so John and Morgan, I'm going to go ahead and do the first attendance word so you can get a little extra credit. So it's not an official attendance word, but again, it's an extra attendance word. And the attendance word is scooter.
Morning.
Good morning. So again, John and Morgan, when you send in your attendance word, if you send that in, that's going to be an extra word for you. Of course, if you're watching the video, so not if you're here live or online live, but if you're watching this video at a later date, that first word of scooter is not extra credit. It is just required for you. And as a reminder, you have to, if you're watching the video, so not John or Morgan or anybody who's here right now, but if you're watching the video, you need to send, also include a screenshot of when I say the word. So, yeah, nothing new, but some people are still messing it up. All right. So, Chili Ride, where'd you ride from?
Damn.
Where?
Excuse me.
Oh, it's okay.
I had workouts this morning.
Oh, okay.
Yeah.
That's good. At least you're probably awake by the time you get here because you have workouts first. I think that's why they did that to us in boot camp. You know, we get us going right away. So then by the time we were learning stuff, we were awake.
Mm-hmm.
Physically exhausted, but
Right.
awake. We were always physically exhausted. What's this? Let's see. Any plans for the weekend?
Get my life back on track.
Oh, man, okay.
I just seen I didn't even take exam 2.
Oh, there you go. Well, time to do it.
Yeah.
You're not the only one. This is why this is the last semester I'm doing this. So this is the first semester. Maybe it was the last one. If it's not the first one, this is at least one of the first semesters where I made attendance not mandatory.
This is the
It's
first one.
just extra credit. And it's the worst I've ever seen. It's the worst labs, worst exams, worst independent work.
I know I could have joined over Zoom, but I just got back to school yesterday.
Okay. Oh, I don't mean just you specifically.
Yeah, I know.
I just mean in general. Yeah.
I just know I haven't communicated anything with you. So, you know.
Well, the videos are there, so you can go get caught up for any days that you missed.
Thank you.
Let's see.
Am I still able to take that exam?
Yeah, everybody is. Yeah. Now the catch is, it's going to be your overall grade. Well, I'll explain it. When I do the announcements, I'll explain it. I don't want to have to do it twice.
I thought you did it already. My bad.
That's okay. I mean, I did it the previous day. I'll do it again today. All right, we've got less than a minute. So since we're recording, even though attendance is easy, I'm going to say it out loud so it's recorded. Bangura is not here. Casebolt is not here. Tristan is not here. Covington Griggs is here. Devere is not here. Dow is not here. Hughes is not here. Levy is not here. Little is not here. Mankins is not here. Mathis is not here. Rains is not here. Reed is not here. Renee or Elise is not here. Thompson's not here. Andy Dewey's not here. And Elkins is not here. So since Covington Griggs is the only one physically here, I'm going to give him double points. Let's jump into it. As a reminder, If you're watching this live right now, if you're on Google Meets with us, then you have to send in the attendance words, and that's it. For the rest of you, if you're watching the video at a later date, you have to send in the attendance words and a screenshot of when I said the attendance words. So basically, everything as usual, but I'm still repeating it because people are still messing it up. My recommendation is maybe you consider coming into class. I was just talking to somebody else, with one of the students who's physically here, and I was mentioning this is the worst semester I've ever seen for exam scores, pre-labs, labs, you name it. It's been the worst. And this is like I was saying to him, it's one of the first times, if not the first time. I think this might be the first semester where I didn't make attendance mandatory, and it's really showing. So anyway, if you're struggling, I recommend coming to class, or I'm preaching to the choir for those who are online, because at least you're online. But anyway, let's jump into it. I'm not going to do the announcements yet, or the reminders, because I know how that goes. People are going to come in late. So we'll start off where we left off, which was talking about family pedigrees. We started talking about family pedigrees, but we didn't have a chance to get into it. So the first word for attendance, and this is going to be on the screen, so I'm not going to usually ask a question. I am asking a question. I haven't verbally said the answer. Let's see, how am I going to put this? No, I won't do that yet. Let's just move forward. So again, we're talking about family pedigrees, right? So one thing we want to talk about before we even mention family pedigrees is the fact that dominant does not necessarily mean that it's more common. And you know this already because we did the lab. Like think about cleft chins, for example. The whole class, except one person, did not have cleft chins. Cleft chins are dominant, right? So that's a good example of how just because it's dominant doesn't mean it's more common. So there's a word for that, and the word that you need to know for the exam is wild type. So a wild type trait is one that is seen most in nature. It's not necessarily dominant, it's just the one that's seen the most. For example, we have a few students in here now, and I can't tell for sure because I need glasses. But if I see correctly, nobody in this room, including myself, has freckles. But freckles are a dominant trait. So again, dominant doesn't mean it's more common. This will be really a lot more important later when we start talking about some diseases. But yeah,
What does dominant mean?
Dominant, good question. Dominant means, right, so you have two different versions of a gene. Those are called alleles. So in your, each one of your cells for any gene, you've got two versions, two alleles. Sometimes you have the dominant allele, the dominant version, or some you might have the recessive version. If you have the dominant version, the dominant allele, that means its version is the one that overpowers the other one. And it's always demonstrated with a capital letter, and I'll just use A as an example. So if you might have a big A and a big A, two dominant alleles. That means whatever that gene is for, you're going to show the dominant version of that. Or you might have a dominant allele and a recessive allele, in which case you're still going to show the dominant version of it because the dominant overpowers the recessive version. Does that make sense? Now, if you have two recessive alleles, little a in this example, then you would show the recessive version of that gene. So using chin dimples again as an example, of course, technically it's the letter D instead of an A. If you were homozygous dominant like this, you would have the chin dimple because that's a dominant trait. If you were heterozygous, where you have a dominant and a recessive, you would have that chin dimple because it's a dominant trait. The only way you would have a smooth chin is if both of your versions of that gene, both of your alleles were recessive, then you would show the recessive version. Makes sense. And that's why they call it dominant, because that one dominates over the other one. So good question. Any other questions about this slide? All right, so the first word for attendance is, as usual, me asking you a question. What do you call a trait that is seen most in nature? So it's not necessarily specified by a dominant allele, but it is the trait that you see most in nature. What is that called? It's a three-letter answer. A three-word answer. Two of those words are hyphenated. And again, to remind you guys who are here, you don't have to send the words in, but you can if you want to get extra credit. If you're online right now, you have to send the words in before 9 a.m.
to get the regular credit and to be counted as present. And of course, if you're online, if you're watching the video at a later date, you need to send the words and a screenshot of what it looks like when I said the words. Any questions about that? All right, so we've talked a lot about genetics, right? Especially with Gregor Mendel, when we talked about the pea plants and this color flower versus that color flower. And he's like, all right, well, I'm gonna make this flower with that flower and see what happens. But how do we do that with humans, right? Because that's an issue. If you're going to study human genetics, it's not like I could be like, all right, I really want to see more about this gene. I want you to have sex with you, and then let me have your kids, and I'm going to take your kids and make them have sex with those kids, and then, once they grow up, right? You can't do that. That's not practical. It's unethical. Also, it takes a while before humans can reproduce, right? So it's not like you're going to wait around until they could. So that obviously poses a challenge. So how do we do it? So let's talk about really quickly what geneticists do. What they do is they analyze the results of matings that have already occurred, right? So again, it's unethical to force humans to mate so you can study the genetics, right? So you just you go back in time and look at the matings that have already occurred. You assemble all this information into a family tree, and that family tree is called a pedigree. So I'm pretty sure you're already familiar with a family tree. That's nothing new. And you might already be familiar with a pedigree, but basically, but when you think of a pedigree, you should think of a family tree, just like, it's my aunts, my uncles, my cousins, whatever. But in this case, we're not just looking at who's related to whom, but we're also looking at a specific gene, right? Because this is genetics. This can be used to determine if a trait is sex-linked, amongst other things. But I'm going to use that one as an example. I'll talk about that later. But you can use the pedigree for a lot of things. And I might have at least one pedigree on the exam. I'm not sure yet. But at least if I have time, I'm going to come up with some practice problems you can do. So let's do this really quickly. So this is a pedigree for attached earlobes. So remind me, attached earlobes, if I remember correctly, is recessive. And what was it? Free hanging earlobes were dominant. Does that sound right? You guys remember from the lab? Anyway, let's take a look. This is how this will work. And I'm going to take a little time with this. I apologize, but again, you might have one of these on the exam. So anybody with an attached earlobe, and hold on, let me back up a step because I want one of you guys to answer this. Again, free hanging earlobe is a dominant trait, right? Attached earlobe is a recessive trait, and we use the letters E for that. Remember? For that lab, it was the letter E. So it's either big E or little e, right? So if attached earlobes are a recessive trait, then what must the genotype of these purple people be? The ones in purple who have attached earlobes, what must their genotype be? And you can either give it to me in the letters or you can describe it. It's lefty. What is it?
Small a's.
Yes, small, right? In other words, two little ones, right? Little e, little e. In other words, homozygous recessive, right? They would have to be homozygous recessive in order to have that recessive trait. So we can go ahead and fill that in. We know these people are little e, right? So, okay. Now, if somebody has a free hanging earlobe, we don't necessarily know their entire genotype, but we know at least one of them, right? So what would at least one of their letters be? Big, thanks, whoever said it, right? At least one has to be big because that's the dominant phenotype, right? So we know this person's at least a big E something. We know this person's at least a big E something. This person's big E something, right? Anybody with a dominant phenotype, we know they must have at least one dominant allele. I think I'm going to do that under. I'm running out of room. Yeah, let's do that under. Okay, knowing that, let's look at this very top left. How do I put this? All right, let's do some examples, right? So we're trying to figure out the genotype of these two people. So we can do some Punnett squares to figure it out. We'll do some what-if Punnett squares. So I know for a fact that two of their kids have at least, how do I put this? No, I don't want to waste your time. I'll just make this quick. we can see for a fact that these two kids are homozygous recessive, right? So that means they have how many recessive alleles? Yeah, they have two, right? So they get one allele from one parent and one allele from the other parent. So therefore, what must the rest of this genotype be? Big E and what? What is it?
Heterozygous?
Yes, it has to be, right? Each one of these parents has to have at least one small e because two of their kids got two small e's, right? So that means they got one from the mom, one from the dad. Therefore, their parents must be heterozygous to give them that recessive allele. Now, this one we don't know about. This person right here, we don't know if that's a big E, or big E, little e, and there's no way to figure it out. Now, we could, do this for the parents here. We know they're both heterozygous, and we know if I did that and worked out that Punnett square, we know there's a 1:3:1 ratio. So I would know that there's a 25% chance, and don't let this frustrate you if you don't know how I came up with these numbers. I can explain later if you want, I just don't want to waste your time now. I know there's a 25% chance that that person's big E, and there's a 50% chance that person is big E, little e. And again, I'm not going to spend time explaining that because this isn't that important. This is a 100 level class. There might be one question about it on the exam. And if you forget this, you'll still be okay later in the semester. You know I'm big on that, right? So you really need to know some of this stuff because you can't understand later stuff unless you know it. This isn't one of those things. So I'll leave this at that. Yes, sir.
So if someone has a dominant trait, does he, if he has just one heterozygous, in his genotype is heterozygous, does that mean in all the other traits is heterozygous as well, or?
Good question. You mean if we're talking about chin dimple versus, all right, though you said heterozygous for chin dimple, is he also heterozygous for other stuff like freckles? Mm-hmm. No, great question. Absolutely not. That goes back to a very important concept that we were talking about previously, independent assortment, right? These genes have nothing to do with each other. There's a slight caveat to that statement, but I'm not even going to say it because it's still theoretical and we don't teach it yet, but you can look it up for independent work if you want. I'll just leave it at that. But yeah, they have nothing to do with each other. For example, I don't know. You could be homozygous recessive for one trait and homozygous dominant for another and heterozygous for another. They're not related to each other. Great question. And that is independent assortment. But anyway, yeah, so I'm going to leave this like this. If I have time, I'm going to try to come up with some problems that you can do for extra credit and come up with a whole worksheet that you can fill out. In which case, I would do one of these problems and actually go through each single one of them and also show you how I got that 25% and that 50%. But for now, we'll just leave it at that. Any questions about pedigree? All right. So the next thing we're going to talk about is human disorders controlled by a single gene. So here we are. It's basically the same thing. We're still talking about dominant versus recessive, and here's your genotype, and here's your phenotype. Except now, instead of talking about freckles and chin dimples, we're going to be talking about diseases. Before we do that, let's get into the reminders. So again, exam 2 is still open for retake. It'll stay open until exam 3 closes. So the retake is not timed. The retake is open book. You can do it as many times as you want. The only catch is you have to email me when you're done so I know to go in there and average your first grade and your highest grade. Without you telling me, I won't know to do it. That also means you can meet me online for exam 1 to get all the correct answers for exam 1 so you can get your 5% boost to that grade and so you have all the correct answers to help you study for the final exam. I'll also remind you, Lab 8, the human genetics lab that we did today is due today. Make sure you get that turned in. Technically, the pre-lab for next week is due today too, but this is one of those things where I really don't. As long as you get it done before Wednesday, you'll be fine because it's like a 3-question pre-lab and it's all hypotheses. And you guys are going to be working on your own, so you're not screwing over your classmates if you're not prepared for lab on Wednesday. Anyway, your lab report. Thanks to Lebdi, I think, pointed this out, was supposed to be due last Wednesday because that was 2 weeks after the lab. So then I pushed it back to Monday because last Wednesday was spring break. But according to Moodle, it's always been Friday, this Friday, today. So I stuck with that date. So lab report's due today. Everything that you need to do the lab report can be found in the announcements. Go to the announcements and look for that one announcement that says something like photosynthesis respiration lab. Everything you need is there. Click on it. It's got the data, videos, everything you need. The last day to drop this class is sometime in this month. I forget exactly when. You should look into it. Some of you should really, really look into it. I'm telling you, you have two choices. Some of you doing fine, but you should either drop this class or start coming to class and have me help you. I'm here to help you. It's a lot of work to turn it around, but I can help you do it if you want to do it. Don't wait until the last week of class to do it. That won't work. Speaking of which, we've got about 8 weeks left to get 100 points worth of independent work. And again, to remind you, just turn in papers. It could be a 2-sentence paper. It could be a 2-page paper. Doesn't matter, right? Just turn in papers. Let me help you if you want. And also, once again, as a reminder, the Friday before and the Monday after Easter, we will not physically be meeting here. We will be meeting online for lecture and office hours. So, before we move forward, my next word for attendance is to ask you about how many weeks do you have left to get 100 points of independent work? How many weeks do you have left? And again, if you're watching the video at a later date, you need to send the word and the screenshot. Any questions about this slide? All right, now let's jump back into it. Human disorders controlled by a single gene. So some disorders are known to be dominant or recessive and controlled by a single gene, and they're also located on your autosomes. These are the ones we're going to be talking about. So again, dominant and recessive. This is exactly what we've been talking about, right? If you have the dominant allele, you have at least one, then you have the dominant phenotype. In this case, the phenotype would be a disease. If you have the homozygous recessive genotype, right? So if you have 2 recessive alleles, then you'll have the recessive phenotype, in this case, a disease. Also, again, like it says right here, the ones we're talking about, these are not sex-linked things. So these are not on your sex chromosomes. That will be important later. There are some exceptions. When we start talking about sex chromosomes later in this chapter, that will be important. But for now, this is basically just pointing out everything we're talking about now is not about sex chromosomes. Also, some things about this first bullet point. I haven't told you this yet, but so far, everything I've taught you about dominant versus recessive, and this is just one gene controlling these things, this is actually not normally the case. So we're teaching this, I'm teaching you this because this is the simplified version of things. And later in this chapter, you're gonna realize, oh, it's not actually usually that simple. Usually there is more than one gene, or usually it's not as simple as dominant versus recessive. We'll get to that later, but for now, I just want to point out that these diseases we were talking about, it's just like everything else we've talked about so far. Dominant versus recessive, one gene controlling it. Here's a table. I don't remember if this table is from your textbook or my previous textbook. This is good for, well, I guess, I was going to say it's good for studying, but it's really not, because I'm not going to ask you anything about these diseases. But yeah, here's some recessive disorders like albinism, cystic fibrosis, PKU, sickle cell disease. We'll talk a lot about sickle cell disease in the next few chapters. That'll be a big one. Not because it's necessarily that important, but it's a great disease to use for different examples, and you'll see what I mean when we get there. But anyway, you can read all about that if you want. We're going to talk about some of these in the next few slides. So to remind you, before we even jump into this, I'm not going to ask you any questions about these on the exam necessarily. Like, there's nothing to memorize, but I will give you one example. There's going to be a question about achondroplasia, but it's not like you need to memorize anything I'm about to teach you about that disease. Because for that question, I'm going to give you all the relevant information you need to know. And it'll be like, all right, if this person with achondroplasia mates with this person without achondroplasia, what are the chances that they will have an offspring with this or with that, right? So all the relevant information you're going to need in the exam, I will give to you. So again, when you're studying, you don't need to memorize anything about these diseases. You don't even need to memorize, for example, the fact that achondroplasia is dominant and cystic fibrosis is recessive. You don't need to memorize any of that. That will be given to you. So again, we're talking about stuff that's basically like what you've already learned about, which is dominant versus recessive, right? So first, we're going to learn about recessive disorders, which obviously means they would have to be homozygous recessive to have that disorder, right? They would have to have 2 recessive alleles. Afterwards, we'll learn about some dominant disorders, which is where you can be homozygous dominant or heterozygous, right? As long as you have that one dominant allele, you're going to have that dominant disease. So let's jump into it. Most disorders are recessive, at least most human disorders. Any guesses why most of these are recessive? No guesses? That's okay. Think about that. And then when we get to the next one, when we get to dominant disorders, then I'll ask you again, why do you think there's fewer dominant disorders? So just let that question marinate for a little bit and we'll come back to it. Individuals with recessive alleles, I need to reword that. Sorry. Sometimes there's people called carriers, right? We're talking about recessive disorders. So again, to have a recessive disorder or any recessive phenotype, you have to be homozygous recessive, right? You have to have 2 recessive alleles. So if you only have 1 recessive allele, right? If you're heterozygous, you're what's called a carrier. And you do need to know that word, carrier. So again, I'm just going to use the letters D, just because that's the next slide. So again, if you're homozygous dominant, you're completely good, right? Not only do you not have the disorder, but you don't even have an allele for it. So you don't have it, there's no way you can pass it along to your offspring. If you're heterozygous, you're fine, right? Because this is a recessive disorder. You don't have both recessive alleles, therefore you're not affected. However, because you do have that recessive allele, you could pass that along to your offspring. Therefore, you are called a carrier. And of course, because this is a recessive disorder, that means you would have to be homozygous recessive. You would have to have 100% of your alleles would have to be recessive for you to have this disorder. So does that second bullet point right here, does this make sense to everybody? Any questions about that? You need to know what a carrier is. So, knowing all this, we can predict the fraction of affected offspring that are likely to result from a mating between 2 carriers. In other words, everything you've already done with these Punnett squares, you can use them and use the same things you've already learned to determine the probability of them having a child with the disorder, or the probability of them having a child, the probability of them having a child with the disorder, or the probability of them having a child that's a carrier, or the probability of them having a child that's just completely unaffected, and that's what this next slide is. So let's assume hearing, there's a single gene involved for hearing, and it's not that simple, this is just an example. If you had 2 carriers mate, right, you can see up here, the mom and the dad are both heterozygous, which means they can hear just fine, but they do have that recessive allele, so there's a chance they could pass it along to their kids. And this should look familiar to you, right? This is that when you mix 3 heterozygotes, you get that 1 to 2 to 1 genotype ratio, right? Same thing, except now we're talking about it in the context of a disorder. So here we go. The chances of these 2 people having a child that is completely unaffected is 25%, right? The chances of these 2 people having a child that's a carrier, that's heterozygous, that's 50%, right? Because you can see there's 2 of them, 2 out of 4, 50%. The chances of them having a deaf child, i.e. homozygous recessive, would be 25%. These are the kind of questions you're going to get on the exam. Not a lot, but you will get a question like this about a disease. And again, all the relevant information, I'll tell you if it's dominant or recessive. And I'll say, this person who can hear mates with this person who cannot hear, or this person who's a carrier mates with this person who's not even a carrier, right? I give you all that relevant information, and then you'll need to do a Punnett square, and then I'll ask you what's the chances of them having a child with this or a child with that. Does that make sense? All right. Speaking of which, the Lab 8, a lot of you didn't get full credit either because you didn't do the pre-lab on time and or you didn't come to lab like you're supposed to. So, and I've gone this and look in the announcements, I've made a new worksheet that you can do where you can get practice doing all these. This will help you on the exam and it'll give you extra credit that can be applied towards your lab.
What can we find?
On the announcements. My most recent announcement, I think, was actually, there's a new announcement that says Lab 8, or Human Genetics Lab. And it says, in this announcement, you'll find everything you need for this lab. So it includes this worksheet, it includes the link So where you upload everything, it's all there. But yeah, I recommend doing this. This would be great, great practice to help you for the exam. And again, extra credit, right? And for some of you, that extra credit will be applied to your lab because you need it. And some of you, your lab will be fine. So that extra credit will just be applied to your semester total. Anyway, any questions about this example? For independent work, here's an independent work idea, and this is odd, I don't normally do this, usually I want you to write something, but for independent work, use this example and tell me what would happen if a homozygous dominant parent mated with a deaf parent. And or what would happen if a homozygous dominant parent mated with a carrier, i.e. heterozygote. Yeah. You can do that for independent work. Tell me the percentages, right? What are the chances of their offspring being deaf? What are the chances of their offspring being deaf? What are the chances of their offspring being a carrier? And what are their chances of their offspring being completely unaffected? Anyway, so that's one disorder. Your book and the old textbook gives an example of cystic fibrosis. This is the most common lethal genetic disease in the United States. And it's caused by a recessive allele that's carried in about 1 in 31 Americans. Put a big old X through this. I only mentioned it because the book did. I'm not going to ask you any questions about cystic fibrosis on the exam. Or at least I'm not going to ask you to memorize this. Again, if I do ask you any questions about cystic fibrosis, I'll give you the information you need, like, oh, this is a recessive disorder. So, I'm not going to ask you anything about how many Americans have it, or what is the most common lethal genetic disease in the United States, but there it is. If you want to, for independent work, though, you can write papers about it. So, for example, it's lethal, right? But how does it kill you? Like, what are the symptoms of the disease? How is it diagnosed? So 1 in every 31 Americans has it? Well, how many worldwide have it? Or what if we get more specific about Americans? What about male versus female? What about African American versus Caucasian versus Asian American versus Latino, right? Things you could look up for independent work. So that's it for recessive disorders. So again, remember, when you get questions about recessive disorders and I say this person mates with this person, what are the chances that their offspring is going to be about that? Just remember, when we're talking about recessive disorders, that you would have to be homozygous recessive to have the disorder. If you are heterozygous, you are a carrier, and if you are completely unaffected, you would be homozygous dominant. And that, you need to know that because in the exam, that's how the wording would be. I would say, like, what happens if this carrier mates with somebody who's completely not affected? Or what happens if this carrier mates with this carrier? Or what happens if this carrier mates with somebody who has the disorder? So know that terminology. Anyway, any questions about recessive disorders before we talk about dominant disorders? Okay, we're going to first just jump into an example. And the first example that we're going to talk about is achondroplasia. Achondroplasia is a form of dwarfism. Before I even jump into it, I'll remind you, just like cystic fibrosis, even though I'm telling you all this information, you don't need to memorize it. Because if there's an exam question about it, I will give you the relevant information. But anyway, I do like this example because it's interesting. The homozygous dominant causes death before birth. So nobody with this, nobody in the world is homozygous dominant for this this disorder because they just don't make it, right? They don't even make it to birth. So it's only, therefore, it's only the heterozygotes that have it. Because think about it. Again, AA, like I just said, ends in death, right? So those people just don't exist. It's a dominant disorder, so heterozygotes, in this case, do have the disorder. The only way to not have the disorder, like everybody in this room, you would have to be homozygous recessive not to have the disorder, because this is a dominant disorder. So before I even move forward, I just want to recognize and point out that this is just an example, and it is an interesting example because the homozygous dominants die. But the rest of most aren't like that. Usually, let's just come up with some other random disorder and use the letter Q. If you have QQ, if you're homozygous dominant, you've got the disease. If you're heterozygous, so big Q, little q, come on, hate this thing. Anyway, then you're heterozygous, you've got the disease. Because you have at least one dominant allele. The only way to not have the allele would be to be homozygous recessive, right? 2 little q's in that example. But anyway, going back to achondroplasia, let's look at some Punnett squares. Again, this is how the exam's some of the questions on the exams are gonna go. It might say something like, what happens when a normal person mates with a dwarf, right? And then you would just need to know, all right, normal person, and again, it will tell you in the question, this is a dominant disorder. So when I say normal, when it says normal person, you're like, all right, if they're normal, that must mean they're homozygous recessive, right? So it won't tell you homozygous recessive. In the exam, you need to know if it's a dominant disorder, and I say they're normal, then they must be homozygous recessive. And then in the exam, if I say, well, in this case for achondroplasia, if they have it, if they're a dwarf, they would have to be heterozygous. Because remember, homozygous dominant would be deadly. And that, again, I will give you that information too. So if there's any questions about achondroplasia, 1, I would remind you that it's a dominant disorder. And 2, I would remind you that homozygous dominant people don't exist because they die before birth. So anyway, if these 2 people mate, again, you do the you can do this Punnett square here and see that their chances of having a child that's a dwarf would be 50%, right? Or 1 in 2, however you want to say it. Their chances of having a normal child would also be 50%, right? It's 50-50. So again, for independent work, I don't normally do this because I normally want you to write stuff, but for independent work, what are the chances of them being a dwarf if you have 2 heterozygous people mating? Or 2 dwarfs mating, right? What are the chances of their kids being a dwarf? What happens if you have a non-dwarf, a dwarf? No, we already have that, mating with non-dwarf. Yeah, I'll just leave it at that. Just the one question. Well, what happens if 2 dwarfs mate? Right? What are the chances of them having a child that's also a dwarf? That's the better question. Even better question. Never mind. I was just going to say, and it's really not professional, and really has not much to do with biology at all. But now that I'm looking at this, I'm just wondering how a non-dwarf person would mate with a dwarf person. I wonder what the mechanics are on that. But that's not too much about biology. That's just how my weird brain works. Anyway, on this slide, for next question for attendance. When a normal person mates with a dwarf, what are the chances, what percent chance is it that their child would be a dwarf? So a normal person and a dwarf mate. What's the percentage, what's the probability of their child being a dwarf? That's your next word for attendance. Or your next number for attendance. All right, so I'm going to ask you this question again before we go to the next slide. Dominant disorders are much less common than recessive disorders. Let me rephrase that, and this is also more of a hint. Deadly dominant disorders are less common than deadly recessive disorders. Anyone want to guess why? Now that we've kind of talked about both. If you're really thinking, and it's a little bit early to be thinking this deep, but if you're really thinking, you should be able to figure out why it would be more common to have a recessive, right? Why? Any guesses?
Because if you have the dominant, you probably already died.
Perfect. Yeah. If you've got the dominant one, you're just gonna die, probably, right? But if you have the recessive, you could be a carrier. You could survive and just be fine, and you're producing kids and just making a bunch of people with that disorder. If you've got the dominant disorder, you're probably gonna die. Probably not gonna pass that gene along. Does that make sense? Mm-hmm. Very good. So yeah, dominant lethal disorders are much less common than lethal recessive disorders. And that is exactly why. Not that I'm necessarily going to test you on that. But I do like this example that both of the textbooks here, the old one and this one, Huntington's disease. So even though I'm not going to question you on this on the exam, but let's quickly talk about it. It's a degeneration of the nervous system that usually doesn't begin until the middle age. So, once you get a bit older, which explains, when you think about it, based on what he correctly said, why that is more common. Huntington's disease is more common because these people aren't usually dead yet, right? They're at least living long enough to procreate. And not only that, I mean, I'm sure they would know these days, but there is a chance someone could have Huntington's disease and not even know it, because like it says in this first bullet point, you don't even start getting symptoms until you're older. So even though that's probably unlikely, I would assume people know that they have it because of parental history and all that. But anyway, yeah, that's something to think about.
So like as you older, like is there something you can go to like to get tested for like everything to see like if
Yeah, you can get tested for these disorders. It's a great question. And you can look that up for independent work because there's all kinds of different ways to do it. And I was just thinking about this, for example, as I'm reading this bullet point, I'm thinking, yeah, most people would know because parents know going into it, like, well, I guess you couldn't be a carrier because you would just have it.
right?
But anyway, I think about like 2 of my daughters are adopted from the foster care system. We don't know about their parents. Their parents may have, I mean, I don't think so, but they may have it, and now we don't know about it. So yeah, to your point, you can test for these things. And again, that's something you can look up for independent work. What do you call it? Like, who do you go to look these things up? And of course, when we're talking about dominant disorders, as we've been mentioning, generally it's not as important because you pretty much know that you have it. You're probably, someone in your family probably died from it. And also, if they had it and it's dominant, then you have it. But yeah, you can look these things up, and it'd be more common for recessive disorders. So yeah, great question and a great topic for independent work. Also, going back to your question about testing, going back to the recessive disorders that we talked about earlier, there is actually a lot of testing for that because they want to know the chances. Like, I'm a carrier, you're a carrier, I know it runs in the family, so let's test our genes before we even have kids. And then you have things like IVF, which is basically artificial insemination, and then you get a bunch of embryos. Sometimes, well, at least it used to be. Laws are changing. But it used to be you might be able to test the embryos to see if any of them have the disorder, and then you find the one that doesn't have the disorder, and that's the embryo that you keep. Anyway, again, I'm going to put a big X to this, reminding you I'm not going to ask you questions about Huntington's disease. Are there any questions about this slide?
How do they call it Huntington's disease?
I actually don't know. I assume it's based off of a person. I used to know, but I forgot. That would be funny, not funny. It would be interesting though if I looked it up. I was like, yeah, it was discovered in Huntington, West Virginia. I doubt that. I feel like I remember that, but who knows? Anyway, the next slides, I kept them in the PowerPoint, even though you really don't need to know anything about them. This is from the old textbook, but some people are into it. It's interesting. So if you want to read like the genetic basis of different coats in dogs, you can read all about that. It's an example of a real study that was done. Really lays out the scientific method, so you can read about it if you want. All right. So going back to your question, genetic testing. There are tests that detect cause of disease-causing alleles. I'm about to scratch the surface on them. But again, for independent work, you can look more into this. I'm not going to ask you any questions about what I'm about to tell you, but it's included in the textbook, and so I'm including it too. Most of these tests are performed during pregnancy if the parents are aware of an increased risk of this disease, like I was saying earlier, especially when it comes to a recessive disease where you could be a carrier, right? So it runs in the family. You might want to get tested. And like this says, usually get tested during pregnancy. Not always, because like I said, sometimes there's IVF. Sometimes people are ahead of the game and like, all right, let's figure out our chances before we decide if we want to have our own kids or if we want to adopt. But anyway, if we're talking about tests that are done during pregnancy, one example is an amniocentesis. This is basically where you pull some liquid out so you can get some cells from the fetus and then take a look at those cells and see what they have. Here's a picture of it happening. It's kind of hard to see because it's bright here. But here, this person's got a very long needle that they're about to stick in. They also have an ultrasound. What do you think they need an ultrasound for? Why do they need an ultrasound when they're sticking the needle in? Yeah, you don't want to poke the baby, right? You just want to get around the baby so you can get some of that fluid that's bathing the baby. But yeah, there's a drawing of it. Again, I'm not going to ask any questions about it. Same with this. There's also something called CVS, where they use a narrow, flexible tube that actually is inserted through the vagina into the uterus, and that removes a little bit of placental tissue. It's less invasive. And then there's also newer genetic screening that involves isolating tiny amounts of fetal cells or DNA that's released into the mother's bloodstream. So sometimes you don't even need to get near there. Sometimes you can just take blood from the mom because some of the cells that are coming off the baby or the fetus are getting into the mom's blood. And, you can take the blood and look for these things. Again, I'm not gonna ask you any questions about this, but it's just so many possibilities to write about the independent work. Like, even if you want to get really specific, like where could I do this? West Virginia. Like if I live in Charleston, West Virginia, and I'm about to have a kid with somebody, where would we go? Which doctor? Anyway. Again, with this genetic testing, once the cells are obtained, they can be screened for genetic diseases. Obviously, patients seeking genetic testing should receive counseling both before and after to explain the test and help them cope with the results. And not just cope with the results, but also you might need to prepare. And depending on the disease, you might need special equipment in your house for this child. You might need special help. Anyway, any questions about that? This is, as far as the exam is concerned, very unimportant. Obviously really important in real life, but not for the exam. All right, so like I mentioned earlier, everything I've taught you so far is a great way to learn about genetics because it's very straightforward. There's this one gene, and this one gene has two different versions of it. And we call those versions alleles. There's either a dominant allele or there's a recessive allele. And the dominant is always, beats out the recessive. Straightforward, right? But now I can tell you that's usually not the case. And what we're about to learn now is stuff called variations on Mendel's law. So again, not always the case. Let's jump into it. So Mendel's laws still do explain the inheritance in terms of genes passed from generation to generation. That part is still true. It still does follow the rules of probability. It is still valid for all sexually reproducing organisms, so not just humans, right? But it does not explain other patterns of inheritance. And for the most part, Mendel's rules cannot account for patterns of inheritance that we usually see. And usually it's more complex. And now we're about to jump into the more complex. The first one we're going to talk about is something called incomplete dominance. So before I do that, let me tell you this. So far, everything I've taught you, and I've not used this word before, but now I'm going to tell you what it is for the exam. Everything I've taught you so far, we're going to call complete dominance. So the chin dimple is an example of complete dominance. If you have at least one dominant allele, you're going to have the chin dimple, right? Same thing with freckles. So if you have that one dominant allele, you're going to have the freckles. That is complete dominance. Now we're going to learn about something called incomplete dominance. And let's see if anyone can guess before I even teach you what it is. So the first example we used for complete dominance was a purple flower and a white flower. You mate them. Remember, they were purebred. Remember, all 100% of their offspring were purple. You guys remember that? So purple and white, you mate, and the rest of all the offspring were purple. That's complete dominance. Now, what happens if we're talking about incomplete, and I haven't taught you yet, so I don't expect you to know this, but we'll see if anybody can guess. What happens if I have a red flower and a white flower, and I mate them? What would you expect their offspring to look like if it's not complete dominance? So I'm telling you now, if you mate the red flower and the white flower in this hypothetical situation, that they will not all be red. So what do you think might happen? Pink. Very good. So here's the definition of incomplete dominance. F1 phenotype is between those of the two parents, which is just the biological, the scientific, academic way of saying what he said, right? If you mix red and white together, right, if you get something between those two, you get pink. So that's what incomplete dominance is. And here you can see these examples. You do not need to memorize these examples, but if you mix, again, white flower or red flower, you get pink flower. Black mouse with a white mouse, you get a gray mouse. Curly hair with straight hair, you get wavy hair. A blue alien mixed with a yellow alien gives you a green alien, right? It's in between. And for the exam, this is how this is going to go. I'm not going to ask you the definition of incomplete dominance. For a lot of this, whether we're talking about complete dominance, which is what we've already talked about, incomplete dominance, which is what we're talking about now, and the other dominance, which we'll talk about later, the questions are going to go like this. I mated this thing with this thing, and these were the results. What is this an example of, right? So if I say I mated a red flower and a white flower, and all the offspring were red, you'd be like, oh, that's complete dominance. If I say I mated a red flower and a white flower, and they were all pink, you can say, oh, that's incomplete dominance. So that's how the exam's gonna go. I'm not going to ask you the definition. I'm going to give you an example of a mating of a cross and give you the results. And based on the results, you need to tell me what kind of offspring they are. But you can see as far as the genotypes are concerned, this is still the same, right? So we have homozygous dominant red flower, homozygous recessive white flower. You cross them, 100% of the offspring are going to be heterozygous. That part's still the same as like everything else I've already showed you. Except now the phenotype is different because now, homozygous dominant is red, right? In this example anyway. Heterozygous is pink. And homozygous recessive is white. Just abbreviate WH, right? So again, the genotypes, that's still the same. Everything I talked to you about Mendel's laws and the laws of segregation and independent assortment, that's all the same. When you could still use everything I've taught you to figure out the genotype ratios. Because again, here we have 1 to 3-to-1 ratio, right? 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So I can still give you this test question and ask you to do a Punnett square and say, what are the chances of their offspring being this? What are the chances of their offspring being that? But in this case, I would tell you first, I'm like, all right, this is incomplete dominance. So what happens if you mix, I don't know, a white flower with a pink flower? I'm not going to do it, but that would be an RR mating with a big R, little r, right? So that's how you might get some questions like that. And again, I would give you all the pertinent information. This is incomplete dominance. Red is dominant to white. What happens when you mix this with this? Or what are the chances of the offspring being this when you mate this with that? All right, the next word for attendance. On this screen, there's 3 generations. There's the P generation, the F1 generation, and the F2 generation. Which of those 3 generations is composed completely of pink flowers? You got that question, right? This is a good way to end it. We've got about 2.5 minutes left, I think. So let's give you a real life example instead of just who cares about flowers, right? There's something called hypercholesterolemia, which means you have way too high blood cholesterol. This is a great human example. It results in dangerously high levels of cholesterol. So dangerous, in fact, that heterozygotes have twice the normal amount. So imagine that, like you have 100% more than you should. Homozygotes, on the other hand, have even more, right? They have 5 times the normal amount. So bad, in fact, that kids as early as the age of 2 can have heart attacks. This is just an example. I'm not going to ask you any questions about it. But here's how it works. It's because this allele, when it's working, so a big H, that allele says, I need you to make this thing, right? Because remember, genes are instructions on how to make stuff, right? How to make proteins. So in this case, if you have both of those alleles, both dominant alleles, then you're making a lot of instructions on how to make these proteins. And these proteins are in your cells, and they're pulling cholesterol out of your bloodstream. Now, if you're heterozygous, you've got one working allele, that big H is working. It says, make this protein, make this protein. But then you've got the little h, which is basically a broken allele. It's a broken gene. It doesn't make any proteins. Therefore, if you're heterozygous, you're still making some of the proteins, but you're not making a lot. Therefore, you're still pulling some cholesterol out, but not a lot. And then, of course, if you're homozygous recessive, you still have the gene, but it's broken. So you're not making any of those proteins. So you're not pulling any cholesterol out of your bloodstream. Therefore, it's deadly. It's very bad. So your last word for attendance, and this is not a word, I'll just remind you what I usually do. The last thing for attendance is to ask me a question about all the material that I covered today, and that's it. We're done for the day. We'll start in with a different type of inheritance patterns on Monday. And don't forget to send me the words before 9 a.m. if you're online. That's it. I'll see you guys either in office hours if you need me or on Monday.